Good afternoon. Thank you for attending our deep dive into Herpetofauna among us, led by our own Julie Wittmann. This is the 12th in the Center for Environmental Inquiry at Sonoma State University's Fall Dig into Nature series of approximately 20 virtual events. My name is Carrie Winninger, and I am the outreach lead with the center. Until COVID-19 hit us, most of our public events were held either at our Osborn Preserve on Sonoma Mountain in Pengrove or at our Galbraith Preserve near Yorkville and Mendocino. And while we are in this pandemic, we are really happy to be able to reach people over Zoom in this new format, which has allowed us to reach a larger, broader audience. So we'd usually pass around a sign-in sheet right now, but in lieu of that, can everyone please take a moment to type their full name into the chat box? We want to make sure your username and your actual name match up. Thank you. And before I let our presenters take it away, I want to tell you a little bit about what the Center for Environmental Inquiry is and how we can be a resource to you, no matter who you are, whether you're affiliated with SSU or not, whether you're a student, parent, government employee, educator, member of the public, or maybe you work with an organization in need of environmental solutions. So the Center for Environmental Inquiry envisions a North Bay working together to find sustainable solutions. And we invite you to come get environmentally ready with us. And we're building a community of problem solvers and learners across all sectors of society, not just the sciences that you might traditionally consider part of this work. And we're providing enhanced understanding of our connection with the environment and skill building experiences that all lead to these sustainable solutions. So there's many ways you can get involved with us. Engage in research is clearly one of them. We have training programs that if you are a student or not, you can be engaged in, such as our naturalist training program and our community naturalist program, of which our leader today, Julie Wittmann, is the coordinator. You can engage with the data that is collected at our preserves. You can partner with us on projects, or you can be a part of events like these. Please do tell us if you have an expertise you'd like to share on an event like this. So the point is that every single one of you is critical in addressing the greatest environmental challenges that we've ever seen. So thank you for being here and for getting involved. So today we're going to talk about the Herpetofauna species found in this area and learn about Julie's research using volunteers to track those. So we'll hear how these creatures can give us some insight into the environmental challenges in our local area and about what we all can do to get involved ourselves, to contribute to research, or to help protect those that are threatened by human action and spread the word. So as I mentioned today, our leader is Julie Wittmann. She is a wildlife biologist, educator, and coordinates the center's community naturalist program, which has actually also gone virtual this fall. So it's been a fun journey. As an SSU grad student herself, she conducted research on local reptile and amphibian species, and she continues to do species counts in the area. So it's just some tech details. This is what we call a deep dive format. What that means is Julie will present about 40 minutes. Then we'll all have about 10 minutes for questions at the end. It's kind of a formal program that will end at 3, but we'll have a few minutes to hang out after to answer any other questions that come up for you guys. Julie has offered to stay a little bit. So what I'm going to do now is I'm going to ask Julie to start sharing her screen, and I'm going to ask all of you to be muted and turn your videos off so that we can really just focus in on Julie and her presentation. And if you can't figure out how to do that yourself, I'll go in a minute and do that for you. You should find it at the lower left-hand part of your screen where you can mute and turn off your video. While we're in the presentation, though, if you have any questions that are technical, like you can't hear her or there's an issue, feel free and put that in the chat box and I can respond. Now, if you have clarifying questions about the content that really would stop you from understanding the presentation, you can also ask that and I'll stop Julie and direct that to her. But most of the questions we're going to stay and keep for the end. And then if anything pops up for you later, if you have questions that come up a week from now, feel free and email me. We're happy to keep engaging on this topic even after the event is over. And in that same vein, this is being recorded today. So it's going to be posted on our website under our past events on our calendar page. And you can also find that just by typing in the URL for this event, which is cei.sonoma.edu/herpetofauna. And you'll see how to spell that on Julie's presentation. And you can always get a hold of me. My email is winingek@sonoma.edu, spelled WININGEK @sonoma.edu. And I'll also put that in the chat. And that's it for me. Julie, take it away.
Thank you, Carrie. As Carrie mentioned, my name is Julie Wittmann, and I'm the Center for Environmental Inquiries Community Naturalist Program Coordinator. I'm also a wildlife biologist in Sonoma and Marin counties, and I'm focused on special status species surveys, especially those involving herpetofauna, which are amphibians and reptiles. I'm also a former Sonoma State University bio-grad student, and as Carrie mentioned, I went through the science teaching credential. As you can see at the bottom of this slide here, I do have some slides where you can contribute your answers or observations, and those are the slides with an asterisk on the upper left. So please do feel free to contribute in the chat box on those slides, or if you have any other questions throughout the program, you're welcome to enter them in the chat box, and I'll be sure to answer them at the end. So first of all, let's start off with amphibians. Amphibians include anurans, which are frogs and toads, and they also include salamanders, including newts, and these other little guys called Sicilians, which are tropical amphibians that look like large worms. We're not going to focus those on today at all, really, because we don't have them. They're tropical. So generally, amphibians are what we call ectothermic. Ecto, Latin for outside of, thermic refers to temperature. Also, the other term that people sometimes use is cold-blooded. So ectothermic just means that the regulation of body temperature depends on external sources such as sunlight or heated rock. So the other thing about amphibians is that they are tetrapod vertebrates. Gotta love that Latin again, tetra meaning four, pod refers to limb, and vertebrates refers to backbone. So obviously, I mentioned Sicilians, they look like these little worm-like things. They don't have legs, but they are still considered tetrapods because in their past ancestry, they did have four limbs, but they have since lost them. So amphibians typically start out as larvae in the water. However, some species have developed behavioral adaptations to bypass this, as you'll see. I'll mention a species or two here today, and you'll see a picture of them. And so also the other thing about amphibians is that the young generally undergo metamorphosis from larvae with gills to adult air breather forms with lungs, generally. Notice the word generally, not always. So amphibians, it's really important to also mention that they use their skin as secondary respiratory surfaces. So, and actually some small terrestrial salamanders and frogs, they don't have lungs, so they rely solely on oxygen exchange from their skin, and some even in the internal lining of their mouths as well. So that's how they survive and get oxygen. And the other really important thing is that amphibians are ecological indicators. So their presence or absence and their general well-being helps us understand our environmental challenges. So they are ecological indicators. Now, reptiles, the other group of herpetofauna include modern critters such as turtles, crocodilians, snakes, and lizards. Extinct reptiles such as dinosaurs, those gave way to birds, and I know some taxonomists groups them with reptiles. So generally, reptiles are, just like amphibians, they are ectothermic. Again, they regulate their temperature using the outside environment, and they are also tetrapod vertebrates. Obviously, snakes, such as the gopher snake on the upper left of the screen here, has no legs, but as I mentioned, and actually there's some lizards without legs as well. But these guys, snakes and certain species of lizards that have no legs, they descended from four-limb ancestors. So we group them with they're tetrapods. They're considered tetrapods. The other thing is reptiles have no aquatic larval stage, unlike many amphibians do, and most are what we call oviparous, another fabulous Latin word. Ove refers to egg, and paris refers to birth. So when something is oviparous, that means that the eggs are laid with little or no embryonic development within the mother. So most, but not all, reptiles are oviparous. The ones that are not are what we call viviparous. That's where the fetus develops within the mother, contained in a placenta rather than an eggshell, or sometimes within an eggshell within the mother. So I'm going to share with you today one aspect to my Sonoma State University Herpetofauna or Herp research focused on amphibians and reptiles. One aspect involved volunteers and technology. So this aspect included specifically determining the accuracy of using a citizen science smartphone app called iNaturalist, used by volunteers to document herpetofauna found during what's called coverboard surveys. More about that later. There's a photo of a coverboard here on this slide, but again, more about that later. But as you can see, I guess it maybe was a coyote or something. You can tell what they thought of my research. So I was going to mention my results of the aspect of this research are published in a scientific peer-reviewed journal, linked on my website, protecthabitat.com. It's also noted on the center of the slide there, and also at the end of this slideshow. But today I'm going to share with you just some aspects of this part of my research, and then also share some information about herps that are found locally. So let me start off by first mentioning the how things worked with my research. So the volunteer and technology aspect of my research occurred December 2015 through July 2016. So it was through a wet through dry season. So it spanned throughout wet and dry months. The volunteers, which at least in the publication I called record contributors, they contributed information or records of data. They had these volunteers that I recruited had no past use of biodiversity smartphone app experience, and these folks, community naturalists and undergraduate volunteers, were recruited through Facebook, preserve newsletters, flyers at Sonoma State, and all they had was a 2.5-hour training to learn the very basics of using the iNaturalist smartphone and website. And so the training just include just very basic overview of the project and use of, like I said, of the citizen science smartphone technology. And it also included one visit to one of the survey sites, which I'll talk about in a little bit. And so all 10 volunteers that were recruited, these human subjects, they all had smartphones and were able to commit to doing 5 surveys over this season. So the natural history mobile app that I mentioned, iNaturalist, it was used, but there's a variety of other apps to choose from, but this is just considered the most standard natural history app, so that's why I used it for my research. There's also another group of volunteers as well that participated in this project. However, I didn't recruit them. These are called, I'm referring to them as identifier respondents. So it's just another type of volunteer, an online volunteer, could be from anybody around the world. And these folks supported the identification of these 10 human subject volunteers by helping them ID their records on iNaturalist. So iNaturalist, the functionality is akin to the Facebook of biology, and that records and information is shared between everybody. So I did want to also mention too, so kind of how some other things work before we start getting into some of the results. But so there would be in the field one volunteer, one rotating volunteer, only one volunteer at a time, and one trained herpetologist, rotating one of two herpetologists that were trained and experienced and completed their bachelor's in herpetology, biology. They recorded the herp data in the field simultaneously. Volunteers used the iNaturalist app, and herpetologists used a field notebook. There were no nets or hands for finding or capturing. It was just simply making observations under a cover board. So, and by the way, the herpetologist lifted the cover board and found the cover boards. So that part wasn't assessed in the study. But first of all, let's kind of, some of you may have heard what a cover board is, some of you never heard of it. So just to kind of reiterate, for folks who may have heard it before, it's, as you can see on the bottom right, that it's a flat piece of material. It's plywood here, but it can be corrugated metal or possibly some other materials. But it's a way to detect terrestrial herpetofauna, and it does provide variations in micro habitat for ectothermic animals. Ectothermic, remember, those animals that regulate their body temperature using the environment. So it also provides protection from predators. These cover boards are also what's called positively bigmotactic, meaning that they like to have things on their back. It also provides habitat for their prey. It's actually something you can set up at your home. I do have cover boards in my backyard, so I've set up several at home here. So I did want to mention also one other thing, a couple things more about this study is that it was a blind study, so the volunteer human subjects were not told about the comparison of the data aspect. So they were just helping out with the project by sharing their data. And then the volunteers, eventually the volunteers' data, I'm just kind of skip down. The volunteers' data was actually eventually compared. So the herpetologist was essentially ground truthing the volunteers' data. And the comparisons included figuring out like what % of the amphibians and reptiles that were visibly seen by the volunteer, and what % of herps escaped during their photograph opportunity. When I say photograph opportunity, let me just share with you some of the volunteer steps. Volunteer, first of all, opens up the app in iNaturalist, which has the camera function, and then they take a photo, one photograph per observation, per organism. Okay, so these are the steps that the volunteers doing in the field. The next thing, the volunteer steps include uploading their observations of the day. Remember they did 5 surveys throughout the span of that time frame. Every single time, then in the evening, they'd upload their observations to the inaturalist.org website. And by the way, that's something everybody here can do. You can join inaturalist.org and contribute as well. It's a great place to check out if you have time. And the other thing too that I wanted to mention, so I did mention how the functionality of iNaturalist is akin to the Facebook of biology, in that the functionality, you could see here on the bottom left side, there's some, people going back and forth about, is it a Western toad? What is it? Blah, blah, blah. So it's just a great way for the global community connect. So once everything is uploaded on iNaturalist, then the identifier respondents, those global volunteers, are allowed to help with the IDing of species that are uploaded to iNaturalist. And then the volunteer iNaturalist data is, like I said, compared to the herpetologist data. And then at last, the accuracy and efforts of the identifier respondents was evaluated. You'll see in that final step, the research grade, that's when enough people actually agreed on a species ID. So then the data, the iNaturalist data, became research grade. This figure is actually in my publication, but I'm just going to briefly go over it here now, and you're welcome to look it up later. The link on my protecthabitat.com website has the link to my publication. But in a nutshell, my research sites were protected areas in Northern California within the Russian River watershed. A total of 144 coverboards, those pieces of plywood, which were half-inch thick plywood, 2 by 4 feet, were set out. However, towards the end of my research, the wildfire that burned through a wildfire, I should say a, now it's not just the wildfire anymore, it's one of the many wildfires, burned through 50% of the plots. So all of these plots that burned were at Pepperwood Preserve. So my 2 protected preserves research sites that I used was Pepperwood Preserve and Osborn Preserve. Osborn Preserve on Sonoma Mountain, that's owned by Sonoma State University, was not impacted, although their fire did go up, I believe it was a couple miles away at the time, but it did not burn at all at Osborn, just Pepperwood. So essentially 50% of these sites were burned. And we're going to zoom in on one of these pond sites in the next slide, so you can see how, just as there's here a total of 4 pond sites between the 2 preserves, 2 pond sites at Pepperwood, 2 pond sites at Osborn Preserve. And then you can see how there are 36 coverboards per site and how they were set up as a function of distance from water, and that they were included in 3 different habitat types, which is oak woodland canopy, oak woodland canopy edge, and then oak woodland forest, and then grassland. So essentially 3 habitat types if you want to throw in that edge habitat. And you can see here the key has the canopy edge, the forest, and the grassland in the figure here. So each coverboard transect was set up in a different habitat type for each pond. And again, protected areas, and 50% of the plots burned. So here that is up close. So this is an example of a pond site and coverboard layout. So this is one of the 4 ponds. This one is actually at Pepperwood before the fire hit. This actually has the actual GPS locations. And you can see that it's within like 3 different habitat types, essentially, these coverboards. And I did want to also add, this is a great time to mention this, that we'll also have another Sonoma State University naturalist program in the spring. We just finished our virtual fall one last Sunday. And where, upon the completion of the program, you can have access to our Sonoma State Osborn Preserve for an inquiry-based project. And so also, as long as the coverboards are not currently being used for research at Osborn Preserve at that time, then you can check them out and flip them up and see what's under there. And the program announcement will be on our Center for Environmental Inquiry calendar, probably by the end of November. But our program will start in February of 2021. And you're welcome to email me if you want me to put you on a reminder list. So there were a total of 18 species of Herpetofauna, amphibians and reptiles, observed before the Tubbs fire hit. And these, again, were all observed by volunteers at Pepperwood only here. There were a total of 3 lizard species, 7 snake species, 2 anuran species, 6 salamander species, including this next species, which is the most exciting find of the volunteers. And here is an asterisk slide. What species is this? It was the first of this species ever observed within Pepperwood Preserve. Feel free again to enter into the chat box if you think you might know the name of this species. So this was a new locality for this species. It was found on the border of its southern distribution, as you see here by the map in pink on the bottom left. It is a plethodontid, lungless salamander, forest salamander, and they do not breed in water, but they do need moist soil. So they are a great ecological indicator species, just like the arboreal salamander, which is the same genus. It has an amazing climbing ability, although this guy's usually, and just like the arboreal, is pretty subterranean. But they have that rounded prehensile tail, and they move fast, just like arboreal salamanders. But again, these guys are mostly just actually entirely terrestrial and don't climb trees very often. But they are red list status, near threatened, and they are declining where they were found. This particular individual was found in Sonoma County, and they're declining due to habitat destruction. And I read that it actually is occurring mostly due to grape vineyards, excuse me. And you could tell the black salamander from the other arboreal salamander. Sometimes their coloration can look a little bit similar, but typically very different. But always look for the rounded toes in the black salamander. So I just told you what it is. So give yourself a pat on the back if you get that it was a black salamander. So that was an exciting find for the volunteers. So many people have gleaned over pepperwood, and to actually just see something that nobody's ever seen before, and through all the JC classes and naturalist programs and everything, for actually just come across a species that wasn't on the preserve list was a really amazing find for those naturalists that put it up on iNaturalist. So I just wanted to kind of go over just briefly a couple non-herp vertebrates that are found under cover boards. They mostly include brush mouse and deer mouse, Peromyscus species, and also California voles. I'll give those species names here. California voles pretty much always in the grassland cover boards. And then less commonly, we've come across shrews and moles. Common invertebrates under the cover boards include ants, spiders, forest scorpions, centipedes, millipedes. Those are all quite common. So never say that when you looked up a cover board, oh, there's nothing under here. There's always something under there. So that wildfire that I mentioned, that was the Tubbs wildfire. It went through Pepperwood, through the research sites, through both pond sites within Pepperwood. This is that wildfire that occurred in October 2017. And it actually grew to become, I think it was like one of the most destructive wildfires in the history of California. But also with all these recent fires, I don't know if we can still say that. But anyhow, this is the preserve burning the night of the Tubbs fire. And large swaths of protected land in the Mayacazus Mountains, where Pepperwood is, and elsewhere burned. Again, Sonoma Mountain did not experience this fire, and just 50% of the total plots burned. And again, this is all at Pepperwood. So really, there's a control aspect for the herp part of the research here. And actually, Julianne Bradbury, Sonoma State University Gurman Group, a grad student, is analyzing Pepperwood, at least Pepperwood's herp data right now. I just talked to her yesterday. So she's going to have some exciting research results soon enough. I'll be sure to try to get those to you when I hear back from her about her final results. So I just wanted to quickly go over some of these awesome herps that was so lucky to see within the first couple surveys immediately following the fire at Pepperwood Preserve at the research site. So the first herps to appear, and they all occurred either under almost completely burned or partially burned cover boards. I still checked them out. I'm a curious naturalist. But it was really exciting to see that some of the first herps to appear. So the cover boards in the forest pretty much burned to a crisp, almost all of it. But some in the canopy edge burned more or less, and then the grassland cover boards just had some burnt corners, essentially. So here's a southern alligator lizard. Tell by the yellow eye and some side markings, but you can't really tell in this photo. But this is next to its burned, presumably its own shedded skin. And this, by the far, was the most severe evidence of any direct impact on herpetofauna immediately, doing those surveys. But there were western fence lizards and western skinks seen. It was kind of a weird place to be right after the fire, but there was still some life, as you can see here. Also, immediately post-fire, there were a few snakes observed within those first, I think it was 4 surveys. And a juvenile racer, as you can see on the left, and a garter snake on the right. And both of those were found in the fire refuge around Turtle Pond. So they I think that Turtle Pond area may have provided somewhat of a refuge for the snakes. So salamander species were also found, such as Encetina, what's called a gummy worm salamander. Frogs and other organisms were also observed under partially burned cover boards, but still no black salamanders yet post-fire to this day. So still, after several years of conducting surveys, still no black salamanders. And this black salamander, like I said, is an ecological indicator And so by just not seeing it, it has really provided us insight into our recent environmental challenges, hasn't it? I'm going to describe this graph here because this is the one slide I'm going to share with you about the citizen scientists, the volunteers, results. How well did they do? So as you can see here, the title is called Detecting Herpetofauna Specimens. And there's, and it's by herp group. So we have the 4 herp groups here, the neurons, the frogs and toads, the salamanders, including newts, the lizards, and the snakes. So the gray part means the percent not detected. So that just means the percent not visibly seen by the volunteers compared to those that were seen by the herpetologist that was with them at that moment in time. And then, so the orange is the percent of the herps that escaped during the photograph opportunity when the volunteer was using the iNaturalist application to document herps. So as you can see here, the salamander group was the least likely to be visibly detected, during the survey. Okay, again, this is not related. This aspect of the study was not related to the iNaturalist citizen science app, but it was just about the inclusion of volunteers to detect salamanders or herps in general. So as you can see, nearly 0 escapes occurred though from the salamander group, and if I remember correctly, it was only 1 California newt that snuck into a rodent hole. But other than that, we didn't have any pretty much 0 escapes that occurred within the salamander group. So now I'm going to ask you a question. As you can see, there's an asterisk on this slide. Which herp group was most likely to escape during photograph opportunity by the volunteer? Which herp group, I'll say it again, see if you can have a moment to enter it in. Which herp group, the neuron, salamander, lizard, or snake, was most likely to escape during photograph opportunity? This is why I love graphs. And if you said the lizard group, give yourself a pat on the back. You are correct. So the salamander group was the least visible to volunteers, and the lizard group was the most likely to escape. And however, I should say something about the salamander not seen issue. The salamanders that were not visibly seen by volunteers, basically almost all of them, were California slender salamanders. So that is, as you can see, it's well, actually, there's no scale on this one, so you wouldn't know that it's a small salamander, but it is a small salamander, but it's a very camouflaged salamander. All the other salamanders were visibly seen. So the only issue for volunteers not seeing salamanders, it was because of the camouflage species. The California slender salamander, are only common small salamander that occurs. However, of course, we have juveniles too, but out of all the salamander species, this is the smallest adult-sized salamander we have at the research sites. Here we go. Western fence lizard that you could see here in the photo, it was the most likely to escape, and actually the southern alligator lizard was the least likely to escape. So skunk was somewhere in between, but the escape during photograph opportunity was really due to additional factors, such as the presence of, especially with western fence lizard, especially due to weather effects. So on cloudless days when temperature was high, volunteers just had trouble documenting. In fact, we got a couple photos of tails and stuff on really hot days, but it was very hard for the global online volunteers to ID those species when you see a little bit of a tail or just not much in the photo. So overall, volunteers were most accurate and very effective for documenting salamanders, other than the slender salamanders, and also they were effective for documenting non-western fence lizards during cooler days, cooler times. And this graph here is also in the publication as well. There are also other herpetofauna among us. Not all of these from here on were found by volunteers during my research. There's some here, but some, such as this species, were actually not even in the study area. And some other species, actually, and also this species, require different survey techniques than cover boards. So here are eggs in the top left corner. A string of, actually, the world record that I saw was 22 eggs in a vernal pool. And this is a California tiger salamander, this species here. So the, actually, I did write that on here. Okay, California tiger salamander here. So here you can see the eggs, larva, and adult California tiger salamander in a vernal pool habitat in western Sonoma County within the Laguna de Santa Rosa. And this species is actually federally endangered and requires permits to survey and handle them. So whenever I do my CTS surveys, I'm always legal to do that. So this is, seriously, there's some major environmental challenges, and right now, and what's really happening to this species is a major decline in this species. So they're impacted by climate change and habitat destruction and alteration, for example, development and other changes in land use. So this is a California tiger salamander with eggs on the upper left, developing larvae in the bottom left, and then hatched larvae from the eggs on the bottom middle, and then the adult, which is odd. There's an extra limb growing out of the rear. That's another conversation, but that is from the Sonoma State University's tunnel project. This is one of the individuals caught during the tunnel project in Cotati area. Here's another species. This is a California red-legged frog. Both of these photos here, California red-legged frog. It's a federally and state-threatened species, another species that requires a permit to survey and handle. And this is our state frog. Yes, this is, again, California red-legged frog. Environmental challenges include, for this guy, includes climate change, habitat destruction and alteration, again, development and other changes in land use. And the frog, actually on the left, was roadkill during a heavy rain incident on Sonoma Mountain. So there's direct impact, there's indirect impact, and the non-native bullfrog, which is not pictured here, but it is a huge challenge for the species. It competes and uses them as prey. So that's another big challenge. And red-legged frog. And actually the photo on the bottom right is taken by Ned, one of our Osborn naturalists, and I believe he took that at Turtle Pond at Osborn Preserve. So here we go, newts. Because that was on the event page, there's a picture of a newt. I wanted to share with everybody what exactly is a newt. As I mentioned, it's a type of salamander. So newts have 3 metamorphoses during their life. They have an aquatic larva. They have a terrestrial juvenile stage, and they have an adult stage. And newts are often more aquatic than other salamanders, although not always. And adults can be fully or semi-aquatic. And all newts, though, breed in water. So you can see here in the upper left corner, there's defensive posturing called the Unken reflex. It's on the top left there, like I said. It's showing its coloration. So this is an aposematic cue, so it serves to warn and repel predators. Newts have tetrodotoxin, TTX for short. This is a neurotoxin, and it's found in their skin and eggs. In Tarica species, all of these are Tarica species. Here we go. There's all the species. So it just that plays. So you could see the defensive. Yeah, I already mentioned that. Okay, so one cluster of eggs is found. I found on the pond edge. So I have the photo up there on the right. If you ever see these washed up on a pond edge and it feels like a bouncy rubber ball, it most likely came from newt. And if it was found in a pond, it most likely came from a California newt, although not always. And if it's more kind of slimy, more likely to be a Nuren frog. Let's see, what else did I want to say? Oh, I did want to mention too about the differences in these 3 species. So these are the 3 species that we have in the North Bay, Sonoma and Marin County specifically. Osborn Preserve has the bottom left California newt and rough-skinned newt, and then Pepperwood, the other research site, has the red-bellied newt. Now, the bottom two left, California newt and rough-skinned newt, were the only ones found under cover boards by volunteers, even though Pepperwood had the red-bellied newt. For some reason, the red-bellied newt never liked to go under cover boards. And as I mentioned, actually, in the naturalist program, the best way to tell the difference between especially the California newt and rough-skinned newt, because they don't have that bright orange, like the red belly does on the top, but is the eyes of the California newt appear to extend to or beyond the outline of the head when viewed from above. So that is the best way to determine their unique characteristic. A lot of people say, oh, California sunset, they got the light lower eyelid. There's reasons why we don't always use that characteristic as defining California newt. Here we go. I'm going to ask you a question. What species of newt is this? Feel free to enter in the chat what species of newt this might be and how can you tell? This one was found at Pepperwood Preserve near one of the pond sites. And a Sonoma State volunteer discovered it. Take another 10 seconds. Feel free to enter into the chat box. What species of Taurica newt might this be, and how can you tell? So it was found at Pepperwood, so that leaves us with three options, three Taurica options, just like on that slideshow that I showed you last time. Last slide. So this is an all dark morph of a California newt. Again, as you can see here, especially on the right photo, actually probably, well, maybe more so on the far right photo, the eyes appear to extend to or beyond the outline of the head when viewed above. So this is a California newt dark morph. Very cool find that Alex Moore from Dr. Gurman's lab documented during his Herpetofauna research at Pepperwood. Okay, so here's another asterisk. Feel free to contribute. This is the larva of which species? And by the way, those two bottom photos, I very gently took them out of the water just for about 5 seconds to take the photo. So these are all in their aquatic form here. This is all the same species. So which species is this? Which species of Taurica newt is this? And big bonus points if you can know the diagnostic feature or features. So yay if you said California newt. This is a larva of a California newt, and it has two, as you can see in the far left photo, actually the best, two dark stripes down the back. It's visible, like I said, from that overhead view. So that's very diagnostic, the two dark stripes down the back. Also, the dorsal fin appears to extend all the way to the shoulders. So again, still all 100% aquatic at this point. So this is a California newt. Here's my question to you guys. Remember I mentioned the neurotoxin that newts have, Taurica species. Here's my question. This was found under cover board. Very exciting situation. Did the frog survive tasting the toxic newt? And by the way, this is called the F stage of a newt. That's the very first terrestrial stage of a newt, terrestrial juvenile phase. So basically an F is like when a rebellious teenager trades gills for lungs. They're really tiny, as you can see here. That's a standard Sierra tree frog. It has a distinct black eyeliner, goth eyeliner through its eyes. And you see my species names up here. I always use iNaturalist names because that's, work with volunteers and we use iNaturalist. So taxonomists will maybe punch me. Some of them might, but that's okay. I just use iNaturalist names. So did the frog survive tasting the toxic newt? I have no idea. I didn't try to keep the frog to see if it would die or not. So I don't know the answer, and I'm sorry I don't have the answer for you, but I'm glad that the frog just did his own life and carried on. So it would be very interesting to know. I did want to mention garter snakes. You know, there are, so basically garter snakes are often considered Tarica newt eaters. So these were also observed by volunteers under cover boards. So in some populations of garter snakes that co-occur with newts, they have evolved resistance to the tetrodotoxin that the newts have, and that allows them to prey on these newts. And this predation has essentially generated like a coevolutionary arms race, I guess, and it's centered around that neurotoxin level in the newts and the resistance in garter snakes. So it's thought that any garter snake that eats a Tarica species of newt is poisonous to predators such as hawks. Though I don't know if that's actually been determined. I did not see it anywhere in Amphibia web, but it would be interesting to learn more about that for sure. Can a hawk survive if it eats a garter snake that just had one Tarica adult or two Tarica adults in the past week? Very interesting concept. So I'm going to play a sound for you. There's an asterisk here, and you're going to tell me what species you think this is. If you guessed California giant salamander, you are correct. That's their vocal sound when they are threatened. And I got it off of Amphibia web because I didn't want to torture one. Plus, they're species of special concern, and it's not a good idea to handle them. So this is, as you can see here, their habitat is on the bottom left. That's Copeland Creek, a great place to see the larva, which is in the middle. This photo is taken by another one of our naturalists that went through our Sonoma State Center for Environmental Inquiry program. So that is a larva. They have those feathery gills. And then on the bottom right, or the entire right screen, that is a metamorphosized adult. And that was the sound of an adult. And they eat other salamanders. They eat invertebrates such as banana slugs. They also eat vertebrates. They would wipe out. In fact, I found a volunteer with me found one of these metamorphosized giant salamanders about 50 feet away from Copeland Creek at Osborn Preserve, wiping out and eating the pinky babies of a Peromyscus mouse. So yeah, that was kind of an interesting find there. Their length can be up to 1 foot. And one of these adults was, as I mentioned, found eating baby mice. So that was definitely the most exciting day for me and that volunteer. So these are California giant salamanders, species of special concern, another indicator species. And finally, again, I just wanted to reiterate my website, protecthabitat.com, where you can go to actually have a curriculum for teachers involving iNaturalists. I have outline of my research project. I have links to my publication and other things that I shared with the Wildlife Society and whatnot. And my iNaturalist username is Protect Habitat. And I have a final way that you can get involved. And again, if you join our naturalist program, you can join our Osborn Preserve iNaturalist.org project. And now, since we're wrapping up here, I'll let Carrie take over. But in the meantime, please feel free to enter your iNaturalist username into the chat box if you already have an iNaturalist an account. Again, mine is Protect Habitat. I will enter that again here so you can follow me. And everyone, Protect.
Awesome. Thank you so much, Julie. If you want to stop sharing your screen, we can all unmute ourselves and have our videos turned on and kind of have a fun little interactive Q&A now. I already do have a few questions that people have asked, but if anybody wants to ask their question out loud, that's also something you can do. So I'm going to go ahead and ask a few that have come in right now. But again, everybody, just feel free and either raise your hand or just start talking or type in the chat, whatever you feel most comfortable doing. And if you're having trouble like unmuting yourself or something, you can type in the chat and I'll try and help you. But we would love to see your faces now if possible. All right. So the first question that you got, Julie, was, do lizards hibernate?
That's a great question. So, you know, torpor, hibernate. My understanding is that they go through more of a torpor period that is seasonal and also happens throughout the day. And I'm just saying this because after looking at so many cover boards, and specifically at Osborn Preserve and Pepperwood Preserve, it was the most commonly observed organism I've seen. On a very cold morning, they are in a torpor state. In a very cold winter, they tend to be more of in a torpor state. You can actually handle, although it wasn't something we regularly did, you can actually handle them. And they just look kind of sleepy. So I haven't heard that they actually they hibernate, but they definitely go through a period of somewhat torpor, just because they are ectotherms and they're not active, you know, during their colder temperature. But I don't think they formally go into a torpor state. But I just think that because they're ectotherms, there's times when they just have reduced heart rate and they're not very active. Yeah, and you know, a great resource that I find myself going to all the time is Herps and Amphibia Web, 2 online great resources.
Would you go ahead and type those in the chat for us? I'm not sure if there's spaces or if they're capitalized, and I'll go ahead and think about the next question here. So this is Maricela. Could we have a design for the cover boards? Do they have a clear place on them? I'm thinking she means maybe to see through.
Oh, I love that. Amphibia Web. Okay, those are, I can't remember if it's .com or .org, but I just posted those. So I have the protocol for the cover boards in terms of the size, the thickness. I have that along with a list of resources for learning about herpetofauna, terrestrial herpetofauna detection using cover boards. I have that all posted on my website. It's actually a specific tab called cover boards. And so you can go on there and you can figure out, okay, what size are people using? Or you can go and research your own, you know, look up your own methods paper. Like some people like to set up plywood because, you know, it attacks kind of more heavy on the amphibians, but also to get, you know, some snakes and reptiles. Other people who are just strictly, you know, into squamates, you know, into lizards and snakes, and then they'll set up corrugated metal. So, but yes, I do have the cover board plans for you on my website. And you're welcome to email me too.
Could you once again type in your website just so it's at the bottom of the chat for everybody?
Please, yes.
All right, I'm going to go through a couple questions really quick, but then we're almost out of time. So the next person asks about, could you please elaborate on that growing an extra limb photo that you had?
Yeah.
You know, I keep asking people about that particular, that day that we were out at the SSU's research site at the tunnels in West Katati. There was one heavy rainy night, and it was Tracy Bain, who was a Gurman Group researcher, and we found 5 of those with the extra limb growing out, and they range from smaller juveniles to larger adults. Well, I know with especially frogs, when amphibians get a parasite in them during oftentimes their larval stage, their lip growth gets whacked out. And so, I've seen three-legged frogs at Pepperwood, and that's not uncommon. But seeing the extra limb out of California tiger salamander, I have not heard a real explanation for that yet. But I did take lots of photos, and I've shared it with folks. And if anyone has any ideas what type of parasite would do that, please feel free to share.
Okay, I think I'm going to ask one more question and then get to a few things I want to say before the event ends, and then we can follow up after that with any other questions people have. Feel free to stay on. So, Kevin says, I've heard that turtles are so ancient that some scientists suggest they could possibly be in their own group, separate from reptiles. What are your thoughts on that?
That's a great question. So my research did not include detecting turtles. Maybe they might like to, actually I just thought of, they actually might like to sit on top of a coverboard if a coverboard would float long enough for them to sit up on top of there. But we have Dr. Geist, who does a lot of Western pond turtle research. He's an SSU professor. He is a great person to ask about the taxonomy of that. I don't know the details about the taxonomy and where they're grouped and when they're grouped, but that's something that I would really like to know. For sure. And in fact, Carrie, is Dr. Geist holding any virtual events?
Not this semester, turtles? No. Okay. But he does, regularly present for the naturalist program and things like that. So
that's a great question. I'm going to write that one down.
All right. Well, let me just say a few things and then we'll get back to some Q&A. So for everyone who does have to leave in a minute here, I just want to say thank you so much for attending. I hope you all learned as much as I did and feel empowered to, go out there and be able to interact and observe these critters and maybe even upload some observations and tell everybody else about what you know and how the interactions between amphibians and reptiles and humans in our environment. So feel free, again, to email me or Julie after the event with any comments, questions, or ideas for future events. So real quickly, this is just one of about 20 virtual events that we have planned through December. So please feel free and check those out at cei.sonoma.edu/calendar. And you do have to register in advance. And spread the word outside the North Bay because you can attend from anywhere. I know there's people on this call from New York and all over the place. So I just want to call your attention to a few events that might be interesting to this group. So in line with today's natural history focus, we have one coming up called Deep Dive Ticks on the Move. And that's on November 5th. That's with UC Davis researcher Emily Pascoe, and she'll talk about her research on the hosts and predators of these small but dangerous critters. Then on November 9th, we have the fourth in a really special series called Live from the Field events. So these are actually part of an NSF-funded collaboration called the Virtual Field. It's just been put together this year during the pandemic. So these events are aimed at undergraduates, but anyone can come, and they showcase research being done literally across the world at 3 different field stations. So after that, there's a Q&A with those researchers. So it's a really exciting opportunity in this case to learn about nutrient cycling in grasslands from researchers in Colorado, New Mexico, and at Buenos Aires University in Argentina. So we have an international crowd, and it's really going to be an exciting one. Then on December 3rd, we have local nature, photography with assistive technology. This is a really rare opportunity. We're so excited to bring it to you to connect people with disabilities or those who are interested in photography access for all in nature to be able to take part in nature photography. So this is going to be led by 2 wonderful people out of Grass Valley, and they can be contacted in advance so that you can arrange with them the kind of assistive technology that you or your friend or loved one might need in order to participate. So please take a look at that and spread the word as well. And then lastly, our final event of the season, December 12th, is the Winter Writing Walk. That's led by Lakin Khan, who used to teach English at SSU, and it's a follow-up to a fall and a spring writing walk that we've held, which just can I say, as a writer, it really feels eye-opening and empowering to be able to use the written word in a way that I didn't think I could, and to connect deeper to nature and then maybe inspire others. So we really hope to see you at some of those events. Please spread the word. And now we'll give it back to Julie for any other questions. And please do just turn your mics on at this point. You know, we want to have a group conversation.
Come on, everyone.
Oh, and thanks for putting all those links in, Julie. I see it.
Oh, yeah. I might have double dipped a few times.
That's okay. Better to have it there. Oh, and thank you everybody for all of the nice comments I'm seeing in the chat. I'm so glad that it's valuable for you all.
So Julie, this is Maricela, and I wanted to say that the reason why I said yellow-legged frog for the Pacific salamander, or the giant salamander, is because I've heard the Pacific giant salamander sound like a barking dog almost.
Yeah.
It does not sound like that. And our yellow-legged frogs, when they croak, they're underneath the water and they make that weird sound that sounds like that you know, because it's underneath the water. So incredible when they're croaking.
It seems like a lot of anurans, frogs and toads, they do the little squeaks and stuff, huh? Yeah.
Yeah.
Do they sound barky to you though, like that giant salamander?
No, that's why I got confused with that giant salamander sound. And my toads make this other sound that goes beep, 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 beep. And I think they're in the water making that sound too.
Are they're toads?
They're toads, Western toads, adults. So, sorry. I'm sorry, they're having a meeting here, so calling to each other, I think.
Yeah, usually when you approach it, like water's edge, well, other than bullfrogs, which are really obnoxious, the sound is usually a plop.
Oh yeah.
You know?
Yeah.
Very rare do we get so fortunate to hear the squeaks and the little barks, you know, of amphibians. Where do you have yellow-legged frog? Which is going to be, what, a threatened species?
It's threatened, yeah. I live on Bechtel Creek in Mendocino County. We have a really nice yellow-legged frog habitat there, yeah.
Yeah, they like the kind of more sunny, unlike the, red-legged frog, but they definitely like the more like sunny rocks and like the meandering stream. I love it. I love yellow-legged frog. It's just amazing how quickly they're declining.
Yeah,
you know? I'm surprised they're not already a threatened species yet, but it's going through the process. That's right.
It is. They will be. Yeah.
Yeah. Well, lucky you, yellow-legged frog.
Yeah.
And Christopher, I think your question got answered that you had typed into the chat earlier about garter snakes. So that's why I didn't bring it up, evolving to resist the toxin in newts.
Yeah, and I wasn't sure if I guess we don't know what happened to the frog, but my guess was that the frog would probably not survive, but who knows?
And you know, Julie, I think
marker.
You're drawing with marker.
Are you drawing a reptile or amphibian?
No.
It's a great way to learn, is to draw what you see in nature.
Very true.
So Julie, you mentioned Dr. Geist earlier, and I think I remember him telling us during a naturalist training session that he did see a frog eat a newt, and then he saw the frog die and the newt crawl the rest of the way out again.
Oh, wow.
Excellent. That is really exciting and amazing in a sad way for the frog. And the newt, right? I mean, the newt died too, because he ate all of it, right? Not just a little piece of tail. But I guess that frog was smart enough to spit it out, huh? Although it did occur during the coverboard lift. So it might have been like that volunteer maybe saved the life of both the newt and the frog. That actually happened during a coverboard lift.
Wow.
I talked to Suzanne today, and she said she saw at Galbraith a garter snake. She couldn't tell which species, probably aquatic garter snake, and it was eating an adult newt. And I'm like, yeah, that's pretty cool. I was just going to talk about, newts and garter snakes today. But but I asked her, if she saw a hawk try to get it, and she didn't see it. I was just trying to get the answer about, the hawk survival rate after they eat, garter snake who's been filling up on Torika. So, yeah.
Food chain.
Are there any other questions
Yeah,
or comments? Or you guys have things to share about herps that you like? I'd love to hear that.
yeah. So, Julie, I have a question about the giant salamander making the sound. That's really interesting. I learned a lot today, and that was one of the things I learned. I had no idea that salamanders vocalize, that they made sounds. Do we know if giant salamanders are doing that like as courtship? Are they talking to each other? Do we know why they make that sound?
They do it. It's my understanding they only do it when they're disturbed.
Okay, so like if they're
Yeah.
having a really bad day. They might.
I mean, I don't do that, but other people I know have done it. And actually, I work up until he just retired actually last month, but I work for somebody that has their handling permit for working with California tiger salamander. And actually, that's one of the things that he does to see if they have those vocals yet, to see, if it's a metamorph,
Yeah.
because that makes a big difference if something has metamorphosized in terms of how you categorize a success of, a vernal pool. And so one of the things he does is he sees if they make vocalizations. And that, along with a few other things, such as, the ability to close eyelids, like, is that considered a metamorph? And he defines that, and there's several other papers that do define those certain features as, yes, that is actually an adult, officially, So I don't really, maybe if they got bit by something that they would probably squeak, they got manipulated by a curious naturalist, then they would also probably bark or squeak too. But yeah, a lot of salamanders do that. And as I can't remember who that was, mentioned, Marcella, yellow-legged frog make squeaks and amazing amphibian sounds, even though they're just little guys, Although those giant salamanders in California tiger salamanders are actually
It's pretty chunky. It's pretty big. Yeah.
yeah.
Thank you.
So, Julie,
Yeah.
what are the other indicators of a full morph of a reptile or an amphibian?
Oh, you mean for at least for the
like
tiger
a salamander.
salamander?
Yeah.
Or- oh, let's see. Their gills have shrunk enough to not being visible, because they have
Right.
the larvae are aquatic, and so it's really those actually there's 4 features, and the eyelid closing and the squeak, vocalization, and it has to in the tail length ratio to, I believe it's tip of its nose to its vent, something like that. It had to do with the ratio of the lengths of its tail to its other parts of its body.
Yeah.
Sorry, I don't know that exact last one, but if I could find that out and share it with you
Thanks.
later, that last one. Yeah.
Great. Well,
Does
if
anyone
anybody, feel free and share, as she said, too, if you have any interesting stories.
Or your favorite
or-
amphibian, your favorite reptile, whatever. Feel free to share.
I just always think of connections to other things. Kevin has taught us during events he's led about sounds other creatures make. And it's so fascinating to, honestly, I'm not that in tune with those, right? I'm more of a plant person. So it's so fascinating learning all these things at events. And now on my nighttime walks, I used to think, that was a spring peeper, what my mom called it when I was little. But what's a spring peeper? Is it a frog? Is it a cricket? And so now being able to compare all of these and with much more of a fine tuning, it's just really fun. You hear and experience things in a different way.
Peeper? Was it a
I
frog?
think it was a frog. Yeah, now that I'm hearing things.
Yeah.
Is your
But
mom from back east, Carrie?
she is.
Okay, guys, spring peepers are related to the chorus frog we have here, the Pseudocris
Got it.
genus, but they call them spring peepers back east. But I know people who've come west often call the Sierra and tree frog or the chorus frog spring peeper.
She's from Ohio.
Well,
Indiana.
Can you hear me, Julie? I have a
Yes.
question. When I was going to UC Santa Cruz, they
Can you
told me
speak up
they-
just a little bit, or maybe I'll turn up my
Speak
volume?
into the microphone more. How's this?
Oh yeah.
when I was at UC Santa Cruz, they used to sell a sweatshirt that had a picture of a banana slug, and it said "No known predators." And I was thinking, is it recent that they've known that the Pacific salamander eats banana slugs, or has that always been known?
I thought it's always been known.
Okay.
You know, you can see photos of a giant salamander eating a banana slug on Amphibia web.
Yeah, and why? I wonder why they would say
Oh,
that.
probably. I think it might be a sports thing. They had to have it fit into their, dialogue.
And so it just kind
It's
of
a sports thing.
Yeah, well, I mean, it was the mascot,
Yeah.
but I'm just surprised because it seems like they clearly do have predators. So it's just weird that they would say that, but I don't know.
I used to know this naturalist who used to kiss banana slugs, but his whole mouth would go numb.
That's
And
the Banana Slug Club. Yeah, I'm totally a part of that.
Are you part of it?
Oh yeah. Oh, and when
Me
I was
too.
teaching, yes, and when I was teaching naturalist training and with the, leading tours more, I used to even tell people if you licked the banana slug, you get to be a board member of the Banana Slug Club.
Ooh.
But anyway, I only did that one time because it really does make you numb and it's kind of disconcerting.
This is the same naturalist that would leave brownie Cliff Bar along the trail and say, the best way to determine the species of scat these are from is you eat it. He'd never tell the kids he was just kidding. So it's the same
That
fun
wasn't me.
naturalist, but the kids loved him. So, but yes, that's great to know you're part of the. I've kind of stayed away from that Banana Club team, Carrie. Is anyone else here part of the Banana Club team?
No, but I feel like I want to be now. Yeah,
Well, we need some darn rain. How about that?
for sure. So
Awesome.
Julie, you asked what our favorite herps were. And
Yeah.
I love the California king snake.
Ah.
It's so beautiful, and they're able to eat poisonous snakes, right? They eat rattlesnakes.
Yeah. My friend who is a herpetophile loves to one of those herpetophiles that likes to grab everything they see, and I'm not one of those kind, but hey, I learned from that. As long as somebody can learn from it, that's okay. But he just does it on his own, and I think this just happened to him last month, and he grabbed just spit out an entire rattlesnake that was bigger than the king snake itself.
Wow.
Like, I just can't believe that. Pretty amazing that they're able to eat. I mean, a lot of people like to have them around their house because then they don't have rattlesnakes around.
Right.
If you have one of those, you don't have rattlesnakes. I don't know what other snake species they eat.
Yeah,
It's
much better.
somewhat related, but I just saw a video of a bobcat and a rattlesnake, and it made me realize how my house cat, how they'll like do this little bat, 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 really fast retraction thing
Yeah.
with their paws. Mine does it for the vacuum cleaner. But this one did that with the snake. And I mean, they were at it for like 5 minutes, and the snake kept biting and the claw and but that's why it's that fast, just because they're faster than a snake. It was really cool. And they did both get away. So it was okay.
What, the vacuum cleaner?
No.
Oh,
And on just my favorite herp is an ensatina.
yes.
Oh, cool.
For the naturalist program, I did another little, just mainly about the herp cover boards at Osborn. But the ensatina, they do look like gummy type critters,
And they're so orange.
The gummy worm salamander.
Uh-huh.
Yeah, they're so amazing. And the females with their big chunky tail. And yeah, I was surprised to see some of those on the first surveys after the fire because, but they are, canopy edge critters. But I can't wait to see Julianne's results, though. That'll be really exciting. We'll have to have her come do a presentation.
Oh, yes, please.
Yeah.
Please put that bug in her ear.
Anyone else have any other favorite herps they want to share? Molly?
Oh, any other, well, I know when in my school, when we did the Kissing Banana Club, the Kissing of the Banana Slug Club, little words, they would always tell you to tuck your lips in when they told kids to kiss it. So you'd go like this. And so I guess that's one reason they made us do that, so that we don't like have too much exposure to the banana slug when they were like initiating kids into the Kiss the Banana Slug club.
I see James practicing actually with his microphone or what with.
Mm-hmm. Now that I'm mentioning it, I'm not sure how I did it in the past. It's actually kind of hard. Yeah, I'm not sure why. I guess, just to keep from the lip numbing kind of, but I'm not sure how that would mitigate it since it's still like exposed to the skin. Who knows?
I want one
Christopher,
of those sweatshirts, those Santa Cruz sweatshirts.
Christopher, does your child have a favorite
Yeah,
reptile or
exactly.
amphibian?
You have a favorite, Isaac? A favorite reptile or amphibian?
I don't have a sweatshirt.
You don't have a sweatshirt?
No.
Oh. He's two. My older one might. He's a little in the dark still. I think for me, the favorite, I'm pretty promiscuous. I like all herps, but I with these two little guys and the shelter in place for ages, I think ringneck snakes and California splendor salamanders, which we find a lot of in our backyard, are both pretty great, and the kids love to see them, and so that's a lot of fun.
Christopher, do you ever find them together? Considering that
I found them under the same rock, and I kind of wonder how long that splendor salamander is going to last when I find them.
maybe he was waiting for a female to lay eggs before he ate it.
Maybe.
I love ringneck snakes. I call them my gateway reptile because
They are.
I feel like a lot of people that are afraid of reptiles can handle a ringneck, and it's a way for them to be like, look, snakes aren't that scary.
So Alessandra, who's been doing the tiger salamander research out at the vernal pools in West Santa Rosa, she sent me a photo during the actual rainy season, the very brief spring/winter we had. The tail, it was like an anomaly of a slender salamander. It was found in one of the pitfall
I
traps.
saw that. It was
you?
so long.
Oh, I showed it to you. It was God. I should share that on my slide next time. Give her credit, of course. But there's so many anomalies. I mean, not just the extra limb on the tiger salamander, but so many other things like that are going on with amphibians, and you have to wonder if it's related to the health of our ecosystem, or is this like normal like parasite issues, or there's
that tail was like how long? Like literally
it was
almost
twice the size of its from nose to vent.
Oh.
So it was at least twice, yeah.
And I remember back in a class in college learning about how human prescription hormones in streams were affecting frogs, and that story, and I'm wondering if it was a name. It started with an A and a Z in it. I can't remember. It's been so long.
Xenoestrogens?
Yeah, what are your
like
thoughts
that.
on that, Julie?
It's been a long time, so I'm wondering what the current state of the field is, because that was 15 years ago.
Mm-hmm.
I don't. Oh, James, I don't know. I mean, I do know that when certain chemicals are abundant in certain waterways that whatever specific animals rely on, it can like alter the gender bias.
Yeah, that's what I think it was talking about.
Yeah, like depending on what it is, it could make it more male or more female, or the temperature change. Obviously, we know that for crocodiles and turtles and stuff, that could make them have more one gender. So maybe with climate change, maybe we're more likely to see,
Yeah, if anyone's interested in that, feel free and take a look at last semester's talk on Western pond turtles by Wendy St. John. She talked about sexual dimorphism and how temperature-dependent sex determination played out in Western pond turtles.
And she probably has the details on the latest on the taxonomy of turtles then too, Kevin, I bet.
Right. Good point. I could ask her. Yeah.
Yeah.
Thank
Where
you.
is everybody? I'm just curious. Where is everybody right now? I'm from Novato here.
I am in New
Oh, it has
York.
different herps. Okay, New York. Okay.
Oh, you're muted, James.
Oh, stand still now.
I'm in Willits, California.
Oh, cool.
I'm in Yorkville, Mendocino.
Oh.
You know that, Maricela, Margot.
We do the one.
I'm in Kentfield, so right next to San Anselmo.
Where are you, Christopher?
Santa Rosa, the east side by Annandale Park. Well, it's because you moved over there. Move back.
And you guys have power today.
We do. We're not in the PG&E PSPSs any. This year we haven't had it out. Last year we had it out 6 times. We'll see.
And then Molly, I think you're in Roanoke Park.
Yeah, I'm in Roanoke Park right now.
And Volkart, I can't remember where you're calling in from. I remember last time you told us, but I'm sorry I've forgotten. Maybe you don't know how to unmute yourself, so I'm going to request to unmute you.
I think he's in Ukiah.
Ah, okay. Yeah, my dad has come in from Maryland, and as I mentioned, we have an event coming up where people will be calling in from Argentina. So it's really fun. Well, I think that unless anyone has anything else they want to say, I'm going to call it a day. Thank Julie again for the amazing information and putting that all together for us.
Yeah.
It really helps
That's
being
really nice,
here.
Julie.
Carrie.
Yeah, thanks for all the
Thank you all so
information.
much, and we hope to see you all again. Please send us any info, ideas, comments, questions.
Bye.